something on gene cloning, right? We spent a couple of lectures on what a cloning vector would look like, what the characteristics of a cloning vector are, and how do you do selection and all that. Then we saw shuttle vectors and we started looking at expression vectors, right? So today's class, let's see if we are able to conclude the expression, gene expression, at least the bacterial part. And then maybe in the final class on Friday, we will probably close up with expression in yeast, insect cells, and in mammalian cells. Okay. So the fundamentals of cloning for in an expression vector remains the same. It doesn't matter whether you're cloning it in prokaryotic or eukaryotic expression vector. All you need to make sure is that whatever you are cloning, it maintains the reading frame, right? I'm hoping that all of you people, all of you students would be remembering what we talked in last semester, when we talked about translation, what reading frame would mean, how many reading frames are there and all that. So, any gene has, 6 reading frames, of which only one is called as an open reading frame. The other reading frames invariably, has lots of these stop codons, and so they are not called as open reading frames. And so your gene, the open reading frame of your gene and the expression vector that you're going to clone, they need to be in sync so that you don't introduce any stop codons either during the process of PCR or during the process of when you do your other manipulations. Okay, so we need to be very careful of that. Okay, so yesterday let me share my slides. So today we will see a short video on recombinant protein expression, maybe towards the end. Let's quickly get started with the slides. We'll start from where we let off yesterday. I'm trying to share my slide. Let me see. Okay, my slides are visible. Should be visible now.
Yeah, so they're visible.
Okay. So this is what we finished yesterday, right? So in these expression host, when we are trying to express in E. coli, right? This E. coli is E. coli that is used for cloning and the E. coli that is used for expression are different strains. Okay, the strains in which you try to do cloning part, if you try to use them for expression, they are not going to work. Okay. Similarly, the one that you're trying to use for expression, people generally don't use those strains for cloning. Okay, so typically strains like DH5 alpha, XL1 blue, JM101, these are strains that are used typically for cloning purposes. Okay, when it comes to expression, depending on the expression that you choose, different kinds of strains are available. Okay, and this PET system, for example, generally uses a strain of E. coli called BL21DE3. We'll come back and look at that in the next this thing. Okay. So as we said yesterday, this strain of E. coli, which is called as BL21DE3, this carries the E. coli this carries in its genomic DNA the T7 RNA polymerase gene, okay, which is under the control of the LAC promoter, okay, and this is followed by the LAC operator sequence, and then this is the structure of that gene that is being shown here. So Unless under normal circumstances, the E. coli RNA polymerase is not able to transcribe this T7 RNA polymerase gene because the repressor is binding. You know, upstream of this T7 RNA polymerase gene, this repressor is binding, and so it prevents the transcription. Okay, so E. coli RNA polymerase cannot transcribe T7 RNA polymerase gene. Okay, because of the presence of this repressor. Okay, the moment you add this IPTG will bind to the repressor and the repression is gone, right? The repressor protein moves away from the promoter region, and then E. coli RNA polymerase can start the transcription of T7 RNA polymerase gene. So this is a specialized E. coli strain which is carrying the T7 RNA polymerase gene in the genome, okay, in its genome under the control of the LAC promoter, LAC UV promoter, which is a synthetic promoter. Okay, so this is a specially genetically engineered strain. Okay, even though, as we said, in the absence of IPTG, the LAC repressor binds to it. Even then, there is a very basal level of transcription. Very few molecules can actually get past. Okay, and so this is called as leaky expression. And then you have very small amounts of T7 RNA polymerase getting formed. And then this T7 RNA polymerase can actually, create a lot of trouble for us. So what people have done is to also express T7 lysozyme. Okay. All of you must be familiar with lysozyme, which is used for, during the lysis of bacterial cell. So this lysozyme also has another property of binding to T7 RNA polymerase, and it prevents the RNA polymerase from binding to the your PET expression vector. Okay, so there are two components in this strain. One is that your genomic DNA carries the T7 RNA polymerase, and then once this T7 RNA polymerase is transcribed and translated, it comes and binds to the T7 promoter, which is what is there in your PET expression vectors. Okay, and then it starts the process of transcribing and the thing gets translated. Okay, the entire process starts only when you have added IPTG to it. Okay, you can add lactose, but typically what happens, lactose will get metabolized, so after a while it will stop, right? The expression will stop. That is why you add IPTGs, because IPTG is a non-hydrolyzable form of allolactose, right? It's a non-hydrolyzable form. So as long as the IPTG is there, the gene will keep on getting induced. Okay. Did I make it clear why we use IPTG rather than lactose? Lactose will get broken down, right? Lactose is a disaccharide. It will get broken down to glucose and galactose. So at some point, the lactose concentration will come down and the LAC repressor will go and bind again. Okay. If you use IPTG, IPTG is a non-hydrolyzable analog. Okay. So typically, IPTG will keep on, it will be kept bind to the repressor and it'll keep on, making sure that this transcription is always on. Okay, that is the reason why people use IPTG rather than lactose. Okay, all right.
Sir, I
Yeah.
have a question. So does this mean that IPTG just stays in the
Oh,
reaction mixture
exactly.
throughout?
Excellent. Very good question. So IPTG remains unchanged. Okay. So once IPTG is bound, it remains unchanged, and that is why it binds the repressor and the repressor goes off. So there is no way that the repressor can come. Of course, you have to understand one thing. As cells grow, the IPTG concentration will fall down. So we generally add sufficient IPTG so that even if the cells grow, okay, the IPTG remains at a certain threshold so that your gene expression remains without any issues. All right, very good question. Okay, any other question?
No, sir, not from my end.
So these are some of the commercial vector families that are typically available. Okay, PET is from a company called Novagen. Okay, it's a T7 promoter based. There are lots of, as I said yesterday, it started with, I remember when we were graduate students, PhD students, we used to have PET up to 21 or something. PET, right from PET 1A, 1B, 1C, all the way to PET21 or something. So there is this 1A, 1B, 1C essentially refers to the reading frame. Okay, essentially refers to the reading frame. And nowadays, I think the PET expression has gone up to 38, 39, 40, something like that. So, the every year they add a new feature, new addition. So Novagen has raked in lots of money just by exploiting this T7 technology. Okay, then you have PJEX, which we saw briefly yesterday. This is sold by Amersham, which was earlier, this was called Pharmacia. They have taken over, and Amersham sells this, and this comes as a GST fusion, glutathione S transferase fusion. Okay, it is generally your gene is expressed downstream of this, so you will have an N-terminal fusion. And this is, unlike T7, this is done by a TRP promoter, which is a hybrid promoter between tryptophan and LAC promoter. Okay, the -10 is from TRP, the no, -35 is from TRP, and -10 is from LAC. So it is still under the control of LAC operon. Okay. Then you have another vector called PQEs. So these are all series, PET 1, 2, 3, 4, like that. It goes up to 38. Similarly, PJEX has 1, 2, 3, 4, 5, 6, all kinds of numbers. Okay. PQE also has several numbers. This is sold by a company called QIAGEN, and the promoter is viral promoter, E. coli viral promoter called T5 promoter. And this carries a series of histidines, okay, in the beginning. Okay, you could choose a vector whether you want a series of histidines in the beginning or you want to have this histidine at the end. The advantage of these things is you can use very easily purify your protein. Okay. And then you have a series of vectors called PBAD from Invitrogen, which uses arabinose promoter. Okay, there are a few useful fusions that are available. Okay, so it becomes a little easy. And then you have PMAL from New England Biolabs, which exploits the maltose binding protein fusion. This we will see, any from the PMAL one. The last slide is on that. We will spend some time on PET. So it doesn't matter whether you're using PET, PJEX, PQE, or PBAD. The fundamentals remain the same. You need to make sure that you are cloning your gene in frame, okay? And the frame is in sync with the gene of, the gene that is being expressed, okay? So the PET system is the most powerful system which has been developed for cloning and expression of recombinant proteins in E. coli. Most powerful and most widely used one. And target genes are specially cloned in the PET plasmids, which is under the control of a T7 transcription, this thing. And it may have, certain vectors may have translation signals also. Generally, it comes with the optimal translation signal. Okay. The expression is induced by IPTG, and the T7 RNA polymerase is so selective and active that once T7 RNA polymerase comes out in sufficient numbers, the entire machinery of E. coli is hijacked. Okay, so E. coli essentially becomes a zombie. It doesn't, translate or transcribe its own DNA. Instead, now the entire resource is now devoted to the expression of this foreign gene, which is under the control of the in the PET expression vectors. Essentially, in a matter of few hours after induction, literally 50% of your protein can be from your PET expression system. Whatever gene that you have cloned, it can account for almost 50% of the entire protein that is found in E. coli. So it's a very, very powerful and very selective system. And another important benefit of this system is its ability to maintain target genes in the transcriptionally silent state when it is uninduced. As I said, there is always a leaky expression, very small amount of leaky expression in spite of the repressor being there. Okay, but because you have this T7 lysozyme being expressed, it makes sure that even that leaky expression is not harmful to the bacteria. Okay. Target genes are initially cloned in a host that do not contain T7 RNA polymerase gene, and so that essentially eliminates plasmid instability. Many times when we try to clone these things, T7 RNA polymerase, make sure, your plasmids become unstable in those hosts. Okay, that's why I said your cloning host and your expression hosts are different. Okay, and why people don't use expression host for cloning, because many a times your plasmids are unstable. They will get rearranged, they will get all kinds of these things happen in the bacterial cells, so people don't use it. Okay, and two types of T7 promoters are available. And you also have, just as I said, BL21DE3, you have a series of hosts which we will see today, which differ in the stringency of suppressing basal expression levels are available. So it essentially provides a lot of flexibility and the ability to optimize the expression of these genes, different kinds of genes under different conditions. Okay. For the purpose of protein production, a recombinant plasmid is transferred to this equalized strain, which is called as BL21D3, which carries a chromosomal copy of the gene for T7 RNA polymerase, which itself is under the control of LAC-UV5 promoter, followed by the LAC repressor region. And the only promoter which is known to direct the transcription of T7 RNA polymerase in these strains, BL21D3, is the LAC-UV5 promoter. And this promoter is inducible only by IPTG, not by anything else. Okay, so addition of IPTG, I mean, of course, lactose is another this thing, but we don't use lactose. Okay, so the addition of IPTG to growing culture of this BL21D3 induces T7 RNA polymerase, which in turn transcribes the target DNA in your PET plasmid. Okay. So this is shown pictorially here. Okay. What I essentially described is shown pictorially here. So this is the chromosomal DNA, E. coli genome, and this is called as a DE3 lysogen. What is a lysogen? Lysogen is a bacteria, okay, in which the viral genes have been incorporated into the genome of the bacteria. Okay, I'm sure you will all know what a lysogen means, especially in the context of lambda phage. Lambda phage can exist both as a lysogen and as a lytic cycle, right? So a lysogen is something that is incorporated its DNA into the bacterial genome. Okay, so that is why this is called as a lysogen. And so this is the LAC promoter and the T7 RNA polymerase gene. Okay, so this is the E. coli RNA polymerase. So the moment you add IPTG, right, the LAC repression is gone. Okay, and the E. coli RNA polymerase, starts transcribing the T7 RNA polymerase gene. Okay. And once the T7 RNA polymerase gene is under the leaky expression, you can see two things, leaky expression of T7 RNA polymerase, the lysozyme binds to it, and so it prevents the thing from transcribing the PET vector. Okay. On the other hand, when you add IPTG, when there is a full-scale expression of T7 RNA polymerase, this RNA polymerase will essentially start transcribing your gene of interest, which you have cloned into PET expression systems. Did I make it clear?
Yes.
Yes. Yes, sir.
Okay.
So,
Okay, so BL21DE3, this is the workhorse of the T7 system. It carries a lysogenic lambda phage. That is why it is the name of the phage is called DE3. Okay, whoever designated this, they called it as DE3. And so now this strain is called as BL21DE3. DE3. So you have a strain called BL21. Okay. And then you have a strain called BL21DE3. So BL21DE3 is something that carries this lambda phage, okay, which contains T7 RNA polymerase under the control of LAC-UV5 promoter. It's a relatively, wild strain in the sense what we use in the laboratory. It grows fast. There is no difference between things like DH5 alpha or any of those cloning strain versus this strain. It looks pretty much, in every aspect, it looks like those strains, okay. And recent studies have shown that it's a safe strain to work with, in spite of the fact that it carries some foreign genes under its genome. It is unlikely to be pathogenic. So, and there are numerous derivatives of this strain that are available for applications. Okay, and this strain per se is defective in a protease, which is called as outer membrane protease, okay, and another protease called lawn protease. So these proteases generally have this tendency to degrade your foreign proteins, so it doesn't allow foreign proteins to be expressed. And so these strains are specially designed so that these genes are absent. These genes are knocked out of these E. coli strains, okay, both OMPT, outer membrane protease T, and lawn protease, okay. You have several strains of BL21. For example, there is a strain called Rosetta. Okay, now this carries several tRNAs which code for arginine, proline, glycine, leucine, isoleucine, and several things. Then you have another strain called BL21 D3 codon plus. Okay, it carries several additional tRNAs. Okay, typically what happens, remember I keep telling you all students that if you look at Mycobacterium tuberculosis and extremely GC rich, and if you look at malaria, Plasmodium falciparum, it's extremely AT rich. So when you try to express these genes, malaria genes or Mycobacterium tuberculosis genes in bacteria, in E. coli, it runs into codon problem. It runs into a problem called codon usage. So, these additional, these tRNA genes help in overcoming the codon issues. Okay. Then you have another strain called tuner. Okay, which is a lac permease deleted, and you can actually play around with the concentration of IPTG, and you can control the rate of transcription, and so essentially you can control the amount of T7 RNA polymerase. Okay. Now, unlike in these strains, whether it is BL21 D3 or any of these other strains, Rosetta or codon plus, once you add IPTG, it's going to continuously transcribe T7 RNA polymerase. On the other hand, in tuner, you can choose how much the amount of transcription needs to be there, okay, by titrating the IPTG levels, okay. And then you have several other strains. You have salt inducible variant. You have several other variants. Some of these are in the they are recently introduced, okay. So people use it under, not for all this thing, but as I said, BL21D3, Rosetta, and codon plus are now commonly used. Commonly used. Okay, other things are being used. It will take some more time for them to get accepted. Okay. Okay. So expressing your gene of interest. Okay. We have cloned it. We have chosen the PET system. We are, in the process of trying to express this. So what is it that we need to do? An insert for expression. An insert is your gene of fragment, which is called as GOI, or any in this thing. As the vectors that are used for expression work literally essentially carry all the elements that are needed for high level expression. All that we need to do is to create an insert that takes advantage of this design. Okay. And perhaps the most important thing that we need to make sure that the insert is cloned in the correct orientation and in the right translation frame to produce the protein that we are trying to make it. Okay, this is something that is uh, essentially it comes by error. Many a times people look at it, they have a cursory glance, and they will decide, okay, I will clone this vector, and then they realize that the reading frame is not correct. Okay. Now, of course, you have several tools that are available, bioinformatics tools. So you can essentially do a lot of in silico thing before you actually do the real experiment. So that is the big advantage. Okay. Now there are a couple of choices that you have when you try to express your genome fragment. One is not to have any fusion. You just want to express your protein in the native form. Okay, you don't want it to be in fusion with any other protein. Okay, so that is okay. That is perfectly okay. And then there are people who want to, who are new to the field of protein purification. They would like to, take advantage of this thing. So they will try to fuse it with certain proteins so that it becomes easy. It becomes easy to purify this protein. Okay. So you can have either, when you decide that you want to have a fusion protein, the question is, do you want to have this fusion in the N terminal or at the C terminal? Okay, So you can, there are some proteins that are perfectly fine when they have an N-terminal fusion, and there are proteins that cannot tolerate fusion at the N-terminal, but they're okay with C-terminal. And there are proteins that cannot tolerate any fusion. Okay, so you have all these cases available, but most cases, in most cases, most proteins can tolerate the addition of 6 histidine residues. So these are called as hexahistidine residues. 6 continuous stretch of histidine residues either at the beginning or at the end. Okay, so that is perfectly fine. Many proteins have no issues with it. And when you have histidine, so these are called as histidine tagged protein, and it's fairly easy to purify these histidine tagged proteins. Okay, so you can have either no fusion, or you can have an N-terminal fusion, or you can have a C-terminal fusion. It depends. It depends on which application. You may not know beforehand whether which one will work. So you may end up trying all the three. Then one of it will work, and then we will go and run with that system. Okay. So once you have done the cloning, you have made the many preps, and you have verified that the insert is found in the correct this thing, you still need to confirm the correctness of the sequence by sending the sample to a sequencing service, because PCR, even with the most high fidelity polymerase, will create some errors. Okay, and so it is not sensible. It is absolutely not sensible to skip the sequencing process. So even after you have constructed and you have cloned, it is very, very wise decision to send it out for sequencing and make sure that the reading frame is maintained. Okay. Okay. So if you look at N-terminal fusion proteins, for example, when you want to do it as an N-terminal fusion protein, the 5' primer, the PCR forward primer, needs to contain a restriction enzyme site so that you can clone it in the appropriate restriction site in the PET vector, right? It needs to have an ATG codon in frame for the initiation methionine, and it needs to be followed by small amino acids, which could allow for the removal of the sequence, in the end, when we want to purify it. Okay. And then you need to have optimized codons for E. coli. As I said, when you try to express either malaria or tuberculosis genes, you can't really do it in a normal strain. You need to have optimized codon, optimized E. coli strains. Okay. And so this is given as an example here. For example, PET vector, for example, there is an NCO1 site. Okay, NCO1 is CC80GG. This is the recognition site for NCO1. Okay. So when you design your primer, 5' primer, you need to make sure that the beginning of your gene has this CC80GG followed by few other sequences. Okay. So that when you do a restriction digestion, when you digest with NCO1, this can come and it can ligate here, and then this reading frame will be maintained. Okay, so this is for N-terminal fusion. So this just around this, you can have a series of histidine residues. Okay, 6 histidine residues, and you can very easily purify the protein. Maybe in our next class we'll look at that. If it is a C-terminal fusion, for example, your 3' primer, the reverse primer, needs to have the restriction enzyme, okay, and maybe a few additional sequences at the 5' ends for digestion. You need to include your stop codon in frame with the sequence, okay, and we need to use, when we are using the 3' PCR primer, we need to use the reverse complement, not this one. You need to use this as your primer sequence. Are you able to see the cursor moving?
Yes, sir.
Okay, so we need to use this as a primer, okay, and then we need to include stop codon, okay, stop codon at the end of your this thing, okay. Okay, so this essentially gives you an example of purification of a enzyme called human polynucleotide phosphorylase. Okay, so you can actually see that this was cloned in PET28A. Okay, and this has a series of N-terminal histidine tagged this thing. This was expressed as a histidine tagged protein. and the host strain was BL21. Just as I said, DE3, this is called as RIPL, which is another strain of BL21. Okay, and the expression conditions. Look at the expression conditions. It's 18 degrees centigrade. The cells were grown at 18 degrees centigrade for 20 hours, and they were induced with 0.5 millimolar IPTG. So typically, the concentration of IPTG that is used is generally varies anywhere between 0.2 millimolar to up to 1 millimolar. People can use up to 1 millimolar, but in this case, they have used 0.5 millimolar, 500 micromolar, right? So it can range anywhere between 200 micromolar to almost 1 millimolar, but in this case, they have used 500 micromolars. Okay, and you can actually see that, as time progresses, the protein gets expressed at the maximal level, right? And then at some point, it kind of wanes away, right? It is not as good. So you need to harvest your proteins when they are getting expressed at the maximal level. Does that make clear? And this is your flow through. After lysing, they have loaded flow through, they have loaded the wash, and these are different fractions. And you can actually see, so this is uninduced control, okay, where they have not induced with IPTG, okay. And you can actually see that after induction, you can actually see that the protein expression levels are going very high. You may think that this could be this protein. This is probably not this protein. Okay, they have run this thing. When you analyze, you will know that this is probably some other protein that may be co-purifying along with it. Okay, you will see in our next this thing. So when you have histidine tags, in the either at the beginning or at the end, as you can see, this is a histidine tag protein. Okay, you have these special columns that are available for protein purification, which is called as nickel NTA. Okay, nickel nitro. I'll come back to you. Okay, I'll come back to you on this abbreviation. But this is a nickel NTA column, which has this affinity to bind only to histidine tag proteins. Okay, so essentially, when you lyse the cell, every protein will wash off, will go away. Only this nickel tagged protein, only the histidine tagged protein will bind to this nickel column, and then you wash with few more reagents, and then you can essentially purify this protein. So this is typically what has happened. They have washed with 50 millimolar tris and 150 millimolar NaCl. Okay, and you can actually see that this protein, is essentially, done as a purified form, and then they have been able to crystallize this. Okay, you may not be able to see it, but you can actually see few crystals here, few crystals, and then this crystal structure was also solved by X-ray diffraction. Okay, so E. coli is a very fairly nice organism. Okay, but it's not that it is this thing. You still have lots of problems that could be encountered during the process of protein expression studies. You can have either low or no expression at all. You may have very low expression or no expression, and typical reason for that is codon usage. The codons that E. coli uses versus the codons that other organism may use may be different. As I said, E. coli doesn't have those codons that are highly GC rich or highly AT rich, right? So you need to make sure that you are taking care of the codon usage problem, either by using a strain that will supply additional amino acid tRNAs, or by modifying your own gene. Okay, or it could be that the messenger RNA that is produced could be unstable. For some reason, it may be unstable, and so the messenger RNA is getting degraded, or it could be that your protein is not very stable. Sometimes the foreign proteins that are produced, that can be toxic to the cell, and so you may not have enough proteins, so the cells may be dying. Okay, or you could have degradation, or you could have premature termination. Okay, many different things are possible. We need to be aware of that. Of course, nobody knows beforehand, so people end up troubleshooting after they have done the experiment, but that's the nature of the work. Okay, sometimes, because you are expressing at such high levels, 50% of the protein is going to be your protein. Many a times they become insoluble, they form aggregates, and these are called as inclusion bodies. Okay, these are called as inclusion bodies. Okay, so when we have these problems, we need to go back, rework. Sometimes, people end up choosing another system, and then it seems to work. Okay, so it is all, if it is not PET, they may go for PGX. If it is not PGX, they may go for PQE, different vectors, and you will essentially end up expressing it in one or the other system. Okay, many a times people realize that no matter what you use, you may not get a functional protein. And when you don't have a functional protein, it is of no use. So they may end up expressing it either in yeast or in other expression systems. Okay. When you have low or no expression, then the question is, are you sure that the insert is correct and it is in frame? And if it is so, it could simply be that it may be having one or two rare codons, which are probably not used very frequently by E. coli. Okay. And if that is the case, then you need to go back and correct that. Okay. Or you can use strains like Rosetta or Rosetta 2, which carry additional transfer RNAs for some of those things. The question is the protein being degraded? Maybe it is getting expressed, but it is getting degraded. Okay, if it is getting degraded, one simple thing that we can do is to do a Western blot and see whether you see a single band or whether you're seeing multiple bands. When you get a single band, it essentially means that the protein is being expressed intact. When you see multiple bands, that means the protein is getting degraded, right? It is probably being, inside the E. coli, it's getting degraded into several other fragments. Okay. And then do we have the correct expression controls? In the previous one, you saw one without induction. So you need to have appropriate controls to make sure that your expression is correct. Okay. And so let's quickly see this short video. Any questions so far?
No, sir.
Okay. Is the thing visible, the YouTube video?
Yes, sir.
And its expression systems.
Is the audio
Let's
audible?
start with what is protein. Proteins are one of the
Yes,
most
sir, it's audible.
essentials for life. And these proteins perform a vast array of functions within organisms, including catalyzing metabolic reactions, DNA replication, responding to stimuli, and transporting molecules from one location to another. However, when we want to a structure, function, mechanism, pathway, or other facts of these proteins, we will find that the quantities are not sufficient from nature's source. In that case, we need to produce high quantity and purity proteins in a short time and low cost in our lab. Thus, recombinant protein technology is required. Recombinant protein is a mid form of protein, which is generated in various ways to produce large quantities of proteins, modify gene sequences, and manufacture useful commercial products. Recombinant protein is encoded by recombinant DNA, which has been cloned in a system that supports expression of the gene and translation of mRNA. The recombinant DNA, the cDNA sequence of the target protein, is designed to be under the control of a well-characterized promoter and express the target protein within the chosen host cell to achieve high-level protein expression. Modification of the gene by recombinant DNA technology can lead to expression of a mutant protein or a large quantity of protein. Choosing an appropriate protein system is the key to the success of recombinant protein expression. Several factors need to be taken into consideration, including target protein property, intended application, protein yield, and cost. Furthermore, challenges exist for many protein expression projects, especially for large protein, membrane protein, nuclear protein and proteins with heavy post-translational modifications. Nowadays, there are several systems for use. Different systems have different features and applications. Here, we will introduce 4 systems which are commonly used in research and industrial. They are bacteria system, yeast system, baculovirus system, and mammalian system. 1. Bacterian system. Currently, E. coli expression system is the earliest use and most mature expression system. The main method is transfer a vector which inserted target DNA fragment to host cell, and then induce protein expression by a PTG. As the earliest development and the most widely used classical expression system, the E. coli expression system has the advantages of clear genetic background, fast breeding, low cost, high expression, easy purification of the product, good stability, strong anti-pollution ability and wide application range. However, there are many short the prokaryotic expression system. Not all proteins are soluble, and incorrectly folded proteins formed in cytoplasm can form insoluble aggregates, called inclusion bodies, which leads to difficult purification. Moreover, the post-transmodification process of prokaryotic system is imperfect, and the biological activity of the expressed product is low. Thus, other more sophisticated systems are also being developed. Such systems may allow for the expression of proteins previously thought impossible in E. coli. For example, glycosylated proteins. 2. Yeast system. Yeast system as a new exogenous protein expression system, contained merits from both prokaryotic and eukaryotic expression system. It is being widely used in the field of genetic engineering. Complete gene sequence of Saccharomyces cerevisiae are sequenced in 1996. The use of S. cerevisiae in the brewing and bread industry has been known for thousands of years, and is considered to be grassed creatures that do not produce toxins, and has also been recognized by FDA. Thus, the productions expressed from yeast system need not a lot of host safety experiments. However, compared with the new yeast system, S. cerevisiae expression system is not suitable for high-density culture, and it lacks strong and strict regulation promoter. Besides, the secretion efficiency is low. Especially, many target protein which molecular weight greater than 30 kD almost does not secrete. Later, people have developed fission yeast and methanol yeast systems. Among them, the methanol yeast expression system is currently the most widely used yeast expression system. Nowadays, mainly used methanol yeasts are H. polymorpha, Candida bodini and Pichapastris. Pichapastris is the most popular tool. Most all yeast contains the methanoic yeast oxidase gene 1, and the exogenous gene was expressed under the action of the promoter. PAOX1 is a structure with glucose or glycerol as carbon source. The expression of AOX1 gene in methanol yeast was inhibited, and PAOX1 could be activated when methanol was the sole carbon source. Therefore, the expression of AOX1 gene could be increased under the control of the gene. The use of methanol yeast to express exogenous protein production is often up to grams. Compared with S. cerevisiae, its translation is closer to mammalian cells and does not undergo hyperglycosylation. 3. Baculovirus system. The insect expression system is a widely used eukaryotic expression system that has the ability to translate and modify foreign proteins similar to most of the higher eukaryotes. The expression of exogenous protein in the insect cell system by recombinant baculovirus is a more popular expression method. The protein level is up to 1/500 mg per liter. But it is restricted and influenced by many factors such as culture medium, oxygen supply and logarithmic growth and so on. Baculovirus is a large known insect viruses and is the earliest, most studied and applicable insect virus. The baculovirus genome is a single closed circular double-stranded DNA molecule with a size of 80-160 kilobits. Its genome can be replicated and transcribed in insect nuclei. DNA replication is assembled in the bark of the baculovirus, which has a great flexibility and could accommodate large fragments of foreign DNA insert. It is the ideal carrier for the expression of large fragments of DNA. The main advantages of baculostem include 1. Recombinant protein has complete biological function, such as protein-correct folding and disulfide bond. 2. Post-translation modification. 3. High level of expression, up to 50% of the total protein amount. 4. Accommodate large insert protein. 5. Simultaneously express multiple genes. The main drawback is that exogenous protein expression is under the control of the very late viral promoter, where the cells begin to die due to viral infection. Insect expression system is normally used for production of membrane proteins, although the glycosylations may be different from those found in vertebrates. In general, it is safer to use than mammalian virus as it has a limited host range and does not infect vertebrates without modifications. Mammalian expression system. Recombinant proteins expressed in mammalian cell commonly use plasmid transfection and viral vector infection. Stable cell line using plasmid transfection takes several weeks, or even months, while the virus vector can quickly infect cells within a few days. The temporal and spatial differences in protein expression. The expression system can be divided into transient, stable, and induced expression systems. Transient expression system refers at host cell cultures without selection pressure, and exogenous vector gradually lost while cell division. The target protein expression time is short. The advantage of transient expression system is simple and short experimental period. Stable expression system means that the carrier DNA replicate and express long time stably in host cell. Due to the need of select resistance and pressure steps, stable expression is relatively time-consuming and laborious. The mammalian expression system has unique advantage in protein initiation signals, processing, secretion, and glycosylation, and is suitable for expressing intact macromolecules. The foreign protein produced by mammalian cells, which is closer to the native protein, has much more activity than proteins produced by prokaryotic expression system, or by other eukaryotic expression system, such as yeast and insect cells. The disadvantage of the technique is complicated, high requirement, low yield, and sometimes viral infections exist. Above all, there are advantages and disadvantages for all these expression system. The advantage of E. coli and yeast expression system is high expression level, low cost. But their modification systems are different from insect cell and mammalian cells. Protein produced by mammalian cell is similar with native protein. However, the disadvantage is low expression level and complicated operation. The biological activity and immunogenicity of the recombinant proteins produced by different expression systems are sometimes different due to the diverted post-translational processing. Therefore, when considering which expression system to choice, we need take a variety of factors into consideration, such as whether the target protein is toxic, whether the protein need biological activity, whether the glycosylation needed for the protein and cost efficiency, yield, purification, safety and so on. Supported by experienced experimental knowledge and consider all these factors, our expertise will make suitable determination and choice. We protein expression system services in our
All right. So that's company that you can actually clone your gene and they can you express it. They will express it to you for a cost. Okay. So last, I just thought that we should look at this PMAL system, which is from New England Biolabs. Okay, so it comes with a vector, okay, which is driven by the maltose E promoter. It has a multiple cloning site where you can, clone your target gene, and then when you express it, it will express as a fusion protein between maltose binding protein and your target protein, along with a small, this thing that is going to link these two together. Okay, and then when you take this, when you load it on a column which has amylose, okay, this maltose binding protein will preferentially bind to amylose. Okay, so none of the other cellular proteins will bind to it, only this will bind to it. Okay, now you can essentially purify this. Okay, um, okay, there we go. Okay, you can elute with maltose. The moment you add maltose to this, okay, only these proteins are now sticking to the column. This will come out. Okay, and then you can cleave with a specific protease. You can remove your these two, and you can very easily do what is called as a gel filtration column. Depending on what your protein is, you can very easily purify your protein of insert. Okay, and I will just show you an example. Here we go. Okay, so this is your molecular weight marker. So this is an SDS page where they have run the molecular weight marker, and the lane 2 is your uninduced cell, the lane 3 is your induced cells, and your 4, 5 and 6 are the purified one. This is your purified one, and this is where they have done after cleavage, this step, and this is the one where they have been able to purify this target protein very clearly from the separate away from the MBP protein. Okay, so it is something that we need to do experimentally. Many times it looks everything is going fine except that you may not get an expression, so you may not be able to purify. But as we saw in the previous case, human polynucleotide phosphorylase, sometimes people are able to get such a huge amount that they're able to crystallize. When do proteins crystallize? When they're at very high concentrations. Okay, so you are able to produce up to 50% of cells this thing, and by and large E. coli works very well. But as we saw in this slide, where did we go? As we saw in this slide towards the end, okay, so this you can actually see when it comes to cost, the production of your foreign protein E. coli is much cheaper than in mammalian system. Okay, but there are times when you have no other choice, because what you are expressing in E. coli may not even be active, may not even be functional. Now, that being the case, you have to go with the mammalian system or with the insect expression system, and so the cost is going to be very high. There are several proteins that are used in the clinic, for treating human diseases that cost up to $100,000 per the treatment per treatment, and they need to do this for couple of years. So you can imagine that because these proteins are expressed in mammalian cells, and so the cost is very high. There are several other reasons why this cost is very high, but one of the reason is these are being expressed in mammalian systems. Okay, so it all depends on, there is a series of steps that you need to do. As I said before, there is nothing beforehand that you can say this will work in E. coli, this will not work in E. coli. You'll have to try out. You'll have to try out. Only thing is, when your protein is heavily modified, with post-translational modification like glycosylation and all that, then it is a good idea to start with either yeast or insect cell or mammalian cell. People these days now directly go to insect cell. Then if it doesn't work in insect cell, they go to the mammalian cell. But that is only for the most heavily post-translationally modified mammalian proteins. Otherwise, for routine things, E. coli works extremely well. All right. Any questions? Okay, so let's brace ourselves for a quiz in the upcoming classes. All right, I will try to do that. Okay, questions?
No, sir, no questions.
Okay. All right. So if there are no questions, then we'll close this and then we'll meet again on Friday. Okay. Okay. Take care. Bye.
Okay, thank you, sir.